I would like to mention a little bit about the Covaxin, which was produced by the Bharat Biotech, and it is an inactivated virus which is used in India. And there is the AstraZeneca vaccine, which is developed by Oxford, the that is Covishield, that is also a non-replicating virus, and so such kind of viruses are used in different countries. So the RNA virus is the Moderna vaccine. Then there are many other examples for it, like live attenuated vaccines. The whole pathogen is weakened and do not cause any disease but induce an immune response. So it's normally no COVID-19 vaccine has been produced yet as live attenuated vaccines. Mostly the examples are polio, measles, influenza virus, etc. Inactivated virus such as Covaxin, Sinovac, Sinopharm, etc. They're developed by Chinese come under inactivated vaccines. The one that is that we use now, that is Oxford AstraZeneca. It's a viral vectored vaccine. Here a non-pathogenic virus acts as a carrier of the gene sequence of the protein. That's it. Then Pfizer is a RNA vaccine. These are all examples of the recombinant DNA vaccines. There is no licensed vaccines for humans. Few are used for horses and for cancer in dogs. So coming to recombinant DNA, it is more reliable vaccine. Recombinant DNA technology can create more safer and reliable vaccines. Immunologically active and non-infectious agents can be produced by deleting virulent genes. So genes encoding a major antigenic determinant is cloned into a benign carrier organism, like what we said earlier about the AstraZeneca vaccine or the Oxford vaccine. That is, it is a virus vector that is carrying the antigenic determinant. So here that is more reliable vaccine. Genes or portions of the gene that is encoding the antigenic determinants can be cloned into expression vectors, and a large amount of the product purified and used as a subunit and peptide vaccine respectively. So here there is the virulent pathogen with antigens. So these are the antigens. The virulent gene is detected from that, or it is deleted. The harmless attenuated pathogen with altered antigen is used, or it will be the antigen. This is the pathogen, and from that the DNA is isolated, coding for the antigen, and that is administered into an yeast cell, and the cell will synthesize the antigen, and that antigen is procured. So that is about the recombinant DNA type of vaccines. Recombinant antigen virus, that is vaccines, examples are hepatitis B vaccine, another example. So the gene of the surface antigen of the hepatitis B is developed in yeast cells. The antigens accumulate intracellularly in the cells, and this is harvested and purified. So that is about the vaccines. And this is a vaccination schedule of a baby, and from birth they will have that is from 1 month they are first administered with BCG and also HE, that is hepatitis B. Then there is OPV, that is oral polio vaccines, and after 6 weeks again there are booster doses for hepatitis B during the 6 weeks as well as 6 months. Then OPV is again administered or given during the 6 months, 9 months, and DTP it is given for in 6 weeks, 10 weeks and 14 weeks. Then polio vaccines again as administered as injections during the 6 weeks, 10 weeks and 14 weeks. That is normally in the for the babies, and the HIV influenza viruses, that is administered in 3 doses. Rotavirus also you give in 3 doses. Then PCV is given, then MMR is given in 2 doses, and typhoid vaccine is given during the 9 to 12 months, and hepatitis A during the 12 months. And in addition to that, there are other vaccines like varicella viruses, etc. Varicella vaccination.